I was introduced to the term brain when I was 4, when my mother once yelled at me, Anjali, have you lost your brain? Since then, I've been attracted by this beautiful subtle thing more than the gravitational force between sun and earth. And this opportunity gave me a chance to live up my interest. Greetings, everyone. I'm Anjali Sharma, currently pursuing my bachelor's in science in biochemistry from University of Delhi, and will be presenting my little systematic research review done as a part of my summer work. Our brain is constantly changing. We continue to change it throughout our life depending upon how we use it. And right now, if you learn something from this presentation, your brain will change a bit. Imagine you look at a cup of coffee. You see a cup of coffee. But what if, when you looked at the cup, your eyes sent signals to the auditory center of brain? Not only would you see the coffee cup, you would start hearing things too. How distracting! Now imagine how confusing this city would be with all this overlapping sensory information. Well, this might actually happen if we didn't have synaptic pruning, which brings us to the first part of my presentation. What is synaptic pruning? Synaptic pruning is a way to increase the efficiency of connections by removing unwanted synapses. But the first thought you might have is, why would this be a good thing? Don't we need every one of our neurons and synapses? Well, one of the grand strategies nature uses to construct nervous system is to overproduce the neural elements and then prune the excess. During infancy, there is an explosion of synapse formation between neurons by a process called synaptogenesis. Models suggest that learning performance is optimal when synaptic connections are first overgenerated and then pruned. Hence, this period during synaptogenesis is vital for learning and memory function. Explosion of synapses continue till it reaches a peak at about age of 2 or 3. And from that point onwards, brain begins synaptic pruning that can last until late 20s. While the mechanism underlying synaptic pruning is in most parts still a mystery, recent studies have implicated the brain's supportive cell known as glial cell, specifically a type of glia known as microglia are responsible for identifying and removing unnecessary neural connections. A number of signaling molecules are involved in the control of glial cell movement, target recognition, and ingestion. One of them being found through a research done by Beth Steven Group at Harvard Medical School, who found that the CR3 or C3 signaling specific to microglia is involved in the pruning and suggests that engulfment is the underlying mechanism. The synaptic connections that get used less are marked by the C3 receptors, and when the microglial cells detect that mark, they become activated and bond to the protein and prune the synapse. Microglial dysfunction results in behavioral deficits, indicating that microglia are essential for proper brain function. Given the important role of synaptic pruning in sculpturing and refining the brain's neural circuits, it is plausible that aberrant synaptic pruning is associated with a number of neurological disorders such as schizophrenia and epilepsy. As per our review analysis, it was seen that different patterns of dysregulated synaptic pruning have been linked to various neuropsychiatric phenotypes. Too little pruning leaves the brain with too many redundant connections, which can be confusing and inefficient. Excessive synapses are observed in the case of autism spectrum disorder and epilepsy. On the other hand, too, too much pruning results in shortage of connection and is thought to underlie schizophrenia. In the developmental pruning hypothesis of schizophrenia, it is proposed that pruning is disrupted and occurs to an excessive degree in patients. As seen in the graph here, such excessive pruning places the patient below a threshold for the presentation of psychotic symptoms. This disrupted pruning might take a variety of forms. For example, there could be a paucity of synapses beginning in early development, possibly as a result of a pre or perinatal insult or of genetic disruption in early development. Moving to next slide. How does synaptic pruning work? Synaptic pruning is activity driven and follows the use it or lose it rule. To keep it simple, synapses that are rarely used are eliminated, while frequently used synapses are protected from removal. Each time an impulse runs along the nerve pathway, it leaves a little trace in the brain. If we use this pathway frequently, it becomes a data highway. But if the nerves are only rarely used, connections disappear away and brain forgets something once learned. In our review, we came across a research done on rat models which showed that pruning occurred in 80% of synapses, barring the largest and the most used one. Researchers have shown that at puberty, there is an increase in inhibitory GABA receptors, which are target for brain chemicals that quiet down the nerve cells. These GABA receptors triggered the synaptic pruning at puberty in the mouse hippocampus, a brain area involved in learning and memory. Knowing that 80% of the brain development occurs during the first 5 years of life, it's evident that exploring and learning new things does lead to a better brain. So feed your curiosity, guys, and your brain will thank you for it. Further, I would like to draw your attention to this figure here, which shows autonomy and non autonomy of sex specific synaptic pruning. Sex specific wiring in the brain results in dimorphic behavior in the worm's juvenile state. Before they reach sexual maturity, their brain connections are in a hybrid or a mixed state, comprising of both male and female arrangements. As they reach the sexual maturity, their brains undergo a pruning process, which get rid of particular connection and leads to either male or a female pattern. As mentioned previously, keeping in mind the use it or lose it rule, when learning any task, when learning any new task, a proportion of synapses are strengthened with increase of the volume of the dendritic spine. And these large dendritic spine plays important role in performing tasks and so persist for longer time. Meanwhile, the useless dendritic spine or even the whole neuron are eliminated during the learning process. Computational models have suggested that decreasing rate pruning optimizes the construction of efficient and robust distributed networks. Thus, pruning process is an inspiration that suggests effective ways to decide distributed networks across several domains by taking help from the neural networks. Getting back to the main idea, synaptic pruning basically provides our brain with its innate delete button. Although we are only beginning to unravel the ramifications of synaptic pruning in the human brain, this process has significant consequences for normal human brain function and may provide key insights into the cause of some devastating and mysterious neuropsychiatric diseases. It is a critical part of neurobiological development that has tremendous consequences on the fully developed adult. At the end of my talk, I want to extend my thanks to the Beyond Sciences organizers and supporters, and I would like to Thank International Youth Neuroscience Association for the summer school and my teaching assistant for the course, Mr. Max, as he provided me a valuable feedback for my systematic review. Thank you.